keep them as the club, and they'll just use them for judging events.
Whether or not you sell them, it's going to be out of your hands.
That's right. I'll be like, I'll drop them on the ground. So we'll go ahead and start it off here. So I'm Mike Fry. Most of you guys have been to these classes. This is the 4th in our series. There's actually, we set up for 5. So the first one was an introduction, just give you a quick overview of Bjcp and scoring. Score sheets tonight, I don't think we're actually going to score any beers. I just have the score sheets out so you guys can review them. You can take them home. They're yours if you want to keep them. Because we have quite a few beers we're going to get through and some off flavors. The first class was, again, scoring. The second class we did was hops. Then we did water. And then today we're going to do yeast. Kurt's going to talk to us about yeast and fermentation. And our next one is supposed to be November 10th. But because it's San Diego Beer Week, we had to move it to the Thursday the 12th. So I'll put an email out. You don't have to remember all that, but it'll be Thursday the 12th. Hopefully folks can make it. That'll be our last one, which will kind of cover the 4 basics for beer. And then we won't do anything for December because everybody's got the holidays and everything else going on. So we'll try to figure out what we're going to ramp up for the new year. And so there's been talks about doing individual styles, maybe once a week, like, hey, every Thursday meet up if you want to learn about category 1. The next week, category 2, category 3. There's other talks, maybe continuing this or keep going, maybe getting toward the tasting exam. So a lot of you guys, we're kind of geared toward the online exam. So hopefully this is information you guys can use. And without further ado, I'll turn it over to Kurt.
Okay. Thank you. Okay, so I'm Kurt Wittenberg, and I am not a Bjcp judge. I'm here just like you are, trying to learn about the system, but I know a few things about yeast, and I've learned a few more. I actually am a yeast molecular biologist and geneticist, but I don't work on anything having really to do with beer. But I started brewing about, maybe it's 6 years ago now, and I've learned a couple things along the way, and I like to sort of incorporate my knowledge from my work into that, and you'll see a couple of little smatterings of that. But what I'm pretty much sticking to the general knowledge about yeast that is relevant to the brewing community, and I'll try not to geek out about genetics and biochemistry and things like that too much. You folks have all heard this saying, brewers make wort, yeast make beer, and that's sort of true, but you have a little bit of control along the way, as you know, to the yeast part. And what we're looking at here, if you've never seen one of these images before, but this is a scanning electron microscope image of yeast, and you can see down here, there's a guy that is budding, which is how yeast grow, and they make a new cell. So this is initiating the process of cell division. And these little things, interestingly, are bud stars. So they're the place, when this guy detaches as a new cell, he's gonna leave behind this little ring of protein and sugars that sort of stays on the cell. And actually you can tell how old the cell is, how many divisions it's been through by counting those things on the cells. Pretty interesting. So what I've done is I'm going to take you through these topics, and you'll see that a lot of the slides have pictures on them. Pictures aren't all that relevant in many cases to the slide. It's just another picture of yeast this is actually a wild Cerevisiae strain that I isolated from a cider that was fermented by Julian Cider Works. And I just took a picture through a light microscope of it, so you can see a different kind of view of what yeast look like if you haven't looked at them. We've got this is an actively growing culture, so some of these guys have buds on them and they're growing. Other guys just were born. Other ones are going to start making a new one in a bit. So this is sort of the growth phase. So we're going to talk a little bit about what does yeast do and how does it grow, and then we'll talk about the growth processes that happens in your fermenter. And sort of introduce a little bit of what goes right and what goes wrong there. And then we're going to talk quite a bit about fermentation, sort of the process and how we have to control it in order to get the outcome that we want. So that's why I say yeast make beer, but we sort of help them along to get the kind of beer we want. And Well, and because of that, we'll talk about off flavors, and we'll taste a couple off flavors. So we're going to take a break about here, and then we'll get together some off flavors for you to taste. And then I want to go through beer, how you use, how you understand what yeast to use for what style, and how you select strains that are appropriate to your style, what kinds of things you need to think about. And we'll talk a little bit, a tiny bit at the end about other kinds of bugs that are used in brewing. Okay, so so this is one of these sort of gratuitous slides, but I think there's actually some cool, interesting stuff here. This is actually a picture of Carlsberg Brewery I took when I was in Copenhagen many years ago before I brewed. And this is the old brewery that was brewed in built in 1847. And I love this. This was actually a fluorescent sign in town in Copenhagen. It was the sort of the understated Danish personality comes through here. Probably the best beer in town. I think at this point it's probably not the best beer in town, because they have some really great little breweries there. But so these are some views of the brewery and why that's relevant. I'll come apparent in a couple slides. But I want to tell you a few things about yeast. So so yeast is just really a generic term for the unicellular phase of fungal growth. So when fungi grow, they have a multicellular stage, many of them, and many of them have a unicellular stage, and some of them only have one or the other. But if they have a unicellular stage, these things are called yeast. So it's really a generic term. But when we say yeast, we're really talking about baker's or brewer's yeast, which are the same organism, Saccharomyces cerevisiae, or sometimes its close cousin, Saccharomyces pastorianus, which used to be called Carlsbergensis, hence the reason I'm showing you Carlsberg Brewery, and Uvarum. Those were just other names. They're used by brewers a lot of times now still, but they really aren't the name of the yeast. And those are ale yeast and lager yeast. And as you know, sometimes ale yeast are referred to as top fermenting because of their proclivity to float to the top during Krausen, high Krausen stage, and lager yeast as bottom fermenting because they don't tend to do that as much, really is what it comes from. But one thing I want to clarify for people who aren't used to thinking about this stuff is that these are different species of yeast. They're two different organisms. They have very different sets of chromosomes and things like that. When we talk about, Belgian yeast and this yeast and that yeast, they're all Saccharomyces cerevisiae, but they're strains. They're genetically distinct versions of the same, just like you're genetically distinct from me. They're genetically distinct versions of the same organism. And then we know about lots of non-Saccharomyces yeast, for instance, Brett, but many of those are not really desirable to have in your fermentations, and some of them don't even really ferment. So people have selected out bugs that do interesting things that they like to add complexity and stuff. And some of those bugs that add complexity don't really ferment your beer for you. They just add more complex flavors.
I was going to say one thing on those terms of Saccharomyces, those are on the online exams.
Yeah, so you can help in this regard.
Yeah, so
I tried to include lots of information.
So no, that's good. So those were like, they'll ask, the question will be, like the first Saccharomyces cerevisiae. They'll ask you, the question will be like, is this an ale yeast? Is this a lager? Like that'll be some of the questions. They'll also ask about Brettanomyces, and they'll throw the different kind of yeast strains just so that as a Bjcp judge, you understand the differences, but they use those technical terms. Yep. They won't say lager yeast, ale yeast.
Yeah. Okay. And I'm going to apologize also. A lot of my slides are really wordy, but I thought it would be good for everyone to be able to have the slides at the end. They can go back and look at them, and they're all there, and they have a lot of the things I'm saying on them.
When you say they don't ferment, what do you mean by that?
So some of these yeast are not going to go into a wort and make alcohol. These yeast are, but there are some yeast out there that really won't do that or won't do much of that. Like I have a yeast I'll show you later that is a really, really slow fermenter. It takes forever to ferment something out, but it does some interesting things in terms of flavor. So when you do a wild fermentation where you have all kinds of bugs in there, those other things are all contributing stuff, but they may not be helping you get alcohol, but there'll be Saccharomyces in there, or there'll be a Brett or someone like that that can make alcohol.
So they're still eating something and producing something.
Yes, but not necessarily converting a sugar to alcohol. Okay. So what happens when we add yeast to a wort? This is sort of the brewer's perspective. Some people call this the life cycle. That's a complete misnomer. It's not the life cycle. It's how they mate and reproduce and do all that stuff. This is just growth. Okay. So what happens when we add yeast to wort? We talk about this in brewing all the time. First, you have a lag phase. And you always have some little lag phase. And basically, what's a true lag phase is just the yeast adapting to its new environment. You might have not made a starter. You might have made a starter. The starter might have been a 1040 wort that was made with DME, and you might now be throwing it into a 1070 wort that has a bunch of rye in it, and this and that. It just it has to adapt to the new environment in order to do what it needs to do and start growing, start dividing. Okay, so that's the lag phase, and the lag phase involves turning on and off genes and doing all these things that these cells do, and it's highly dependent on the things that are present in the environment, the nutrients, the temperature, the this and that. Okay, but it takes time, and it's generally in the range of hours. If it takes days, you have problems. You start worrying if the next day your fermentation is not bubbling through the airlock. That probably means it's either really slow because these cells were not particularly happy, or because they weren't particularly viable. Hence the reason to use starters. Okay, so you want to have a large viable cell number, and so the amount of cells you have there and the number of viable or living cells is not always the same thing. And if you get a really old batch of yeast that your brewing store had around for too long, a large number of the cells might be dead. And it's always a good idea, unless you're getting a really fresh pack, to do a starter. And obviously there's other reasons to do a starter, and we'll talk a little bit more about that later. But these are just some detailed pictures of those buildings. And up in one of the buildings, the one that's being held up by these elephants, which are really cool, there's this little statue of this couple way up on this thing. And this is the founders of Carlsberg, and those are the Jacobsons, I'm sure they said. So what happens next? So that was the lag phase. You want to keep a short lag phase if you can, and the best way to do that is to get your yeast growing beforehand. Even if you don't do an overnight or a 2-day starter, just put your yeast into some wort or some DME-based wort that you make or something. Get it going. Okay. Then you have what some people call the accelerating phase, or the low Krausen phase. All this means is that growth is now happening, but it's happening slowly because you don't have all that many yeast yet. Okay? Hopefully it's not because 3/4 of your yeast is dead, but it's just because you don't have the full population you're going to have when this thing is really cooking. And this is the initiation. You'll notice that I sometimes say cell proliferation, because when you talk about growth, that could be the cell growing, one cell growing, or it could be the growth of the population, which is what we're really talking about, the cell proliferation. This is going to take hours to a day in this stage to really get going, and it's dependent on many, many things about many of the conditions in the fermenter. Then we're into exponential phase, or high Krausen phase, and you all know what this is if you've brewed. I mean, your wort is all roiling around. You've got yeast and foam on the top. You're cranking out a couple of bubbles through your airlock every second or something. It's really moving along. This can go on depending on largely on the type of beer you're making. Is it a lager where your temperature is really low, or are you doing things in an uncontrolled manner and temperature is really high? The strain, obviously, also the cell number now, which is increasing. etc. So it can go for a long period of time if you're fermenting really slowly. And obviously it depends on your initial pitch rate, because when you have a high cell number, you get going really fast, but it lasts a short time because you eat up all your sugar really quickly or your other nutrients, and then you're heading into the next phase. Things are slowing down. Okay? So people say the optimal pitch is somewhere, a cell number where you get 1 to 3 times, 1 to 3 division cycles, which means 2 to 8-fold increase in yeast mass. Most of us actually, if you think about it, don't pitch at that higher rate. You really have to grow up some liters of pitch if you want to pitch at this rate, and it's a good idea to do it. But this is considered sort of optimal. And another thing, this is dependent on nutrient as well, because if you're low on a particular nutrient, then growth's going to terminate prematurely. You're going to use up the nutrient. Okay, so that's basically accelerating and exponential phase is sort of all exponential phase, and then it starts slowing down, and you have late browsing. Things are going really slowly. It can be relatively short. It can be really long, and you all know about this. Where you're doing things at low temperature, and you're getting a bubble every minute, but it's still fermenting. And then eventually your cells slow down, and you go into what's known as stationary phase or quiescent. The cells start to flocculate, they fall out of solution, and at that stage, they're sort of like you when you're asleep. Things are all slowing down. They're still metabolizing a bit. They can still clean up your beer a bit, which is why lagering's a great thing, and you can leave this to go for a long time, or you can transfer into a secondary and not be sitting on your yeast so much. And part of this, a lot of people don't secondary at all, but a lot of this is dependent on whether you're doing ales or lagers. Whether the lagers, there's much more of a necessity for cleanup, and it also depends on what your style of brewing is. People worry sometimes about autolysis when you're sitting on that bed of yeast, and they're worried about the yeast dying, where you can get these off flavors, which are referred to as yeasty or umami. Or typically, the way most of us do things, unless you go on vacation for 3 months while you're making a beer, this doesn't really get to be a big deal, unless you're really warm or you're lasting or leaving your beer a really long time. Okay, so that's sort of the growth phase. Okay, now the reason I talk about Carlsberg Brewery is that there's a place called Carlsberg Laboratory. It was founded in the late 1800s, and this place is really important for brewing. These guys first isolated brewing yeast as single organisms, so that you could now get this strain and that strain, and that we knew one of them was Saccharomyces cerevisiae and one of them was something else. They were the first lab to isolate, and therefore they named Saccharomyces carlsbergensis, now called Pastorianus. Okay, this is where the whole concept of pH developed. So it's really important in chemistry and biology and all that too. It's where Haldol, if you know the name, first figured out how to measure nitrogen in organic materials and things like that. So a ton of stuff was done here that's really important to brewing and science in general. And I happen to have once stayed in this apartment right here, which is the president's apartment, who they no longer live there. But I gave a seminar here on some work that I did, because there's people doing basic science too. And it was really cool, because they invited me to bring my whole family. We hung out here. We got to wander around the labs. This is actually Kjeldahl's lab from the late 1900s. He was the first president of the lab. And it's got all his old equipment and stuff. It's a very cool place. Anyhow, so they figured out a lot of stuff that has to do with fermentation. Okay, how does fermentation work? And this, I don't want you to even pay too much attention to this, but just to tell you that this line across the top, this is basically a summary of glycolysis, where you take glucose and you turn it into pyruvate. And then the important thing is this pyruvate can go 2 ways. During fermentation, there's no oxygen present. It'll do this. okay? And during oxidative growth, it goes somewhere else. It goes to the mitochondria, and it gets turned into CO2 and water. And we aren't really interested in CO2 and water here. And so this is the reaction we're really interested in. And it goes through acetaldehyde intermediate. And then the other thing to notice that these things, starch is made up of glucose, so is maltose. So this is where we get the glucose to go through this reaction. It actually doesn't get converted directly to glucose, but we'll just pretend this for right now. So the simplified view is that you take starch and you turn it into ethanol and CO2, but it does it by going through simple sugars. As we know, that's why we mash, right? So that this is the process of fermentation. But the other thing to notice is we already have an off flavor, two off flavors here actually. They're part of the process. Ethanol, if there's too much of it, is an off flavor. That's the hot alcohol flavor. And acetaldehyde is the green apple thing that is also an off flavor. So if you have this process is efficient, but this process isn't efficient, you know, the one right here, then you start to build up acetaldehyde. So a lot of things tweak the fermentation process, and that's what we're trying to control. And the thing to think about is that not only, I say fermentation is complex, but that's true. There's a lot more to it than I'm showing. But what's really important is that there's lots of other stuff going on in the cell at the same time. And a lot, you know, the cell's making amino acids to make proteins and things like that, nucleic acids to make DNA and all these things. And some of these things react and form other things we aren't so happy to have, or maybe we want, depending on the beer. But there's a lot of stuff going on here, and there's lots of other sugars around. And so we have to have fermentation control. So I sort of defined it. It's the struggle to maintain the balance between these good reactions and these bad reactions. And some of the bad reactions are actually part of the good reactions. How do we keep this all going the right way? So that's what fermentation control is all about. So I'm going to talk about the elements of fermentation control. What things do we try to control during the fermentation process? Why isn't it just that yeast make beer, but we in yeast make beer? So the first thing is temperature. So every yeast strain has an optimal brewing temperature or brewing temperature range, right? So we know use this yeast at 58 degrees, use this yeast at 68 to 70 degrees, right? But all these yeast, including lager yeast, they'll pretty much grow from, and lager yeast even lower, pretty much grow from 45 or something to 90. And so they'll grow over a huge range. When I grow yeast in the lab, I'm usually growing them somewhere between, let's say 72 and 85 usually, okay? And 85 is sort of a really good temperature for us. We do that. They grow fast, everything's great, but they don't make good beer when you're growing them like that, okay? And they're also not, we're not doing it without oxygen, okay? So if you go to these high temperatures, we try and ferment, 70 degrees or lower. If you go to high temperatures, you can get a lot of these off reactions tend to be favored over the good reactions. And some of the side reactions tend to be favored. So you get esters and fusel alcohols when you're fermenting at high temperature. Sometimes esters are a good thing. Belgian yeast, German Hefeweizen, which we'll taste some of at the end, produce esters from the fermentation byproducts at a higher rate. They're just genetically different. They have some of the enzymes you need to do those things, so they produce a lot of the intermediates. Okay, and as you know, different ones produce different esters. You do one Belgian yeast, it produces things that are kind of spicy. Maybe they're a little phenolic. If you make another Belgian yeast, they're really fruity. You make the banana thing, and then there's a bunch of nasty ones that you don't really want. So there's all kinds of things that go on, and we're trying to deal with this. So a safe way to go is to just sort of decrease your temperatures until you know what you're dealing with. And then with Belgian yeast, when we know what we're dealing with, we sort of let the temperatures rise at the end of the fermentation process, and that gives us more of these esters. So if we have cooler fermentation temperatures, we produce cleaner beers, at least in terms of esters and fusel alcohols and phenols. And so by definition, lager yeast ferment cleaner because they're capable of actually fermenting and growing at lower temperatures than the ale yeast, which sort of grow in this range. So we ferment lagers really cool. They don't produce very many fusel alcohols and esters. They produce a lot of sulfur compounds sometimes, but they don't produce those things. Okay. So that's kind of what we do with temperature. Okay, then another issue is oxygen. So oxygen is kind of this double-sided sword here. We have oxygen is really important because we need to have oxygen for sterile synthesis, which is involved in the synthesis of cell walls. And if you're going to make a yeast divide, you better make it some new cell wall, or it's not going to be very happy. You got to do that when yeast are growing. So you need oxygen to undercover to overcome the lag phase and then start growing. And the optimal oxygen level is sort of 8 to 10 parts per million. And you can introduce that through by pumping air, oxygen, pure oxygen through a fritted stone or a fritted stainless steel that is attached to a rod that you're pumping your oxygen through, so you make lots of really fine bubbles. You can do that with air too. A lot of us, I know most of us that start out just like to shake the carboy really hard, unless you got carboys big enough you can't shake. And we force air in that way. And you can get close to this, but it's pretty hard to get up to this level. But if you do about 30 seconds in a 5-gallon container of a good flow of oxygen with a fritted stone, you can get a really good oxygen level, and your yeast cells will be a lot happier. Okay. Now remember that one of the things that's confusing here is they're still going to be fermenting during this early stage, even though they have lots of oxygen, because there's lots of glucose present at that stage. And there's a thing in yeast, which is a whole other thing that we don't have to go into too much, but it might come up on the exam. I don't know. It's called the Crabtree effect. No. Okay, so the Crabtree effect is just that yeast will ferment even in the presence of oxygen if they have high glucose levels. Okay, they can produce plenty of energy that way, and they're happy doing it even though oxygen is present. But there's a time when you don't want to have oxygen present, and that is later in fermentation, because yeast can use alcohol to produce energy after the glucose and all the other sugars are all gone. And if we let it do that, it needs oxygen to do that. So we don't want oxygen anymore once the fermentation gets really going. Okay, they're happy, they have enough, they've grown enough. Okay, no more oxygen, because now we're going to convert our alcohol into water. Okay, which isn't our goal. Okay, so there's times when oxygen is good, early, and we want a good punch of oxygen in there. We don't want it late. Okay, and the other thing that I didn't mention, but you all probably know, is that oxygen promotes oxidation. Okay, that's its job, and we don't want to have lots of oxidation from environmental oxygen. And then another thing, which I hadn't really heard much about before, but I read a couple things about it, is that if you put too much oxygen in, and this is true of too many nutrients too, you can actually promote too vigorous of growth and lead to off flavors. But I don't think most of us run into that problem. Although I think when you get really aggressive growth early in your fermentations, it's really important to think about temperature control, right? Because you're generating a lot of heat when you're in that stage. The temperature you measure outside isn't necessarily the temperature in the fermenter. So it's good to measure your fermenter's temperature. And then there's two other issues we need to consider. One is pitching rate. So I already mentioned that we want about 1 to 3 rounds of division, or sort of 8-fold increase in cell number during the early phase, the hypralazine phase. What rate that occurs at will be determined by the gravity of the wort, the temperature, the health of the yeast, etc., and the pitch rate. So if you have a low pitch rate, cells grow for a really long time. It's not a good thing. It stresses the yeast. The yeast start producing off flavors. Sometimes the yeast give up too soon, okay? And they lead to poor attenuation. You don't have that big punch of fermenting yeast. You can overpitch, but it's a little less serious but one of the problems with overpitching, I mean, the yeast will still eat up everything that's there. Okay, if you have just way too much yeast, they'll still eat up all the sugars, but they might start flocculating before they get there because there's so many of them. They're so close together and they're trying to sense their environment. They flocculate when they sense things going on in the environment. The nutrients are changing rapidly and they're about to run out. So they might flocculate prematurely and you get poor attenuation, so too much sugar left at the end. And sometimes you'll get autolysis in that condition because the cells get unhappy or your beer sits on your cells for a long time. So you can calculate the pitch rate, and it's best to calculate the pitch rate using a pitch rate calculator. I won't say best, it's easiest. And most of you have probably heard of Mr. Multi. But MrMulti.com has a good calculator and there are other ones that consider more variables, but this one is pretty versatile, and you can get a good estimate of pitch rate. Don't worry if you're 25% off or 25% too much, but if you're fourfold lower, you're adding a lot of stress to the situation. Okay, and some of you are probably making big beers with one vial of White Labs, or at least the old vials where I knew what the number was, but and you really should be using 4 vials worth. You really should have made a starter or spent a lot of money.
One of the feedbacks on judging, and I'll hand out those flaw sheets, is a lot of it is pitch healthy and pitch enough yeast. So that's one of the big feedbacks that you'll have to know when you're going through it. And that'll be part of again, on the online exam, it'll give you examples and it'll have like, if the beer is like this, what are some of the causes? And you have to pick what the causes and feedback that you provide.
Okay, so the other thing, right, is exactly what we're talking about here. You better make a starter. I just want you to look at the starter section because you can grow a starter by just mixing up some DME and some sanitized. Boil up some DME basically. So you have sanitary situation, boil it in your flask or sanitize your flask and boil it elsewhere. Add your yeast, let it grow. You can do that and you can sort of swirl your flask around or a carboy or a bottle or whatever. An important thing is you don't really have to worry about oxygen at this point. It's perfectly fine to grow a starter with just a piece of foil over the top to keep stuff out.
So why is that? That's what I was always curious.
Because actually, you'll get the healthiest yeast that way. You're not trying to make.
So why is it not producing off flavors while it's just sitting there?
Well, it very well may be, but most of us let our pitch settle out. I do, at least.
Okay.
So I cold crash my pitch, or I just let it settle out overnight, and then I pour off the
okay.
more. Because especially if you're doing a couple liters like this, that's a lot of material to put in,
Right.
and you're making it with something that is kind of substandard.
I do the same way. I was always curious, like
Yeah.
why you do that. And the other one was when I do a lager starter.
Yeah.
Why do I do that at room temperature? Shouldn't I do it at lager
Well,
temperatures?
exactly. So we're not. That's
Which
what I'm
one
saying.
is-
If you're letting things settle out, what you really want is healthy yeast. The yeast are happier like this. And the other thing is, the more you can aerate this, the better off you are. So you'll notice it's on a stirring plate. So it's got a stir bar in it, it's whipping around. The best way is if you have something like I do, which is a shaker, a rotary shaker, okay? But all of you can't get one of those. I just happen to have them laying around. So, but the best way is if you could be you when some people just walk by every time they have to think of it. Okay? So if you could sit there the whole time and go like this, you'd be in really great shape, okay? Because you're constantly reducing area.
Yeah, so you'll put it in the fridge and let it cool, and then you'll have the yeast slurry on the bottom, and a lot
So
of people.
then you'll have this big pad of
Condenses
yeast
the
at the bottom.
numbers down huge.
What's
Like
that?
it condenses your yeast numbers down if you want so much.
Doesn't change numbers at all. All it's doing is it's all the yeast are precipitating out, they're flocculating. And especially if you let your yeast grow long enough. And then you don't want to do what I'm talking about if you're just starting it up for 2 hours. Okay, just getting your yeast happy. Just do it in a small volume, get your yeast happy and dump them in. Okay, but if you're really making a 2 or 4 liter starter for a big 10-gallon batch or something like that, then you want to grow up your cells. I put mine in the cold because the cold encourages them to crash out, to flocculate, and you just let them settle. Pretty much you have a clear wort if you have a yeast that flocculates well, and you just dump that stuff off as close to the bottom as you can, and you might be only pitching 250 mils or something like that.
So would you leave it the same when you put it in the refrigerator, or you put an airlock on it?
No.
Okay. Just curious.
But you can put an airlock, and a lot of people do that. You get higher cell density this way. What?
Why would the yeast be happier without adding oxygen? I thought you said
They're happier with oxygen.
you said you don't need to add oxygen. You can just leave it.
But these guys are getting stirred, and we're not doing that with them. You're not doing that with your wort, presumably. If you're stirring your wort, you're doing it like this, right? But these guys, it's getting oxygen from the air. There's turnover, lots of movement. But yeah, you could oxygenate if you really wanted to get into it. I don't know of anyone who does that. Okay.
Yeah.
And then the other issue is nutrition. And it is said, and I read it actually, I was surprised to read it actually, but I read it in Chris White and Jamil's book, that all the nutrients you need are present in a wort except zinc and oxygen. So if you make just a 1040 wort, you're making a low gravity beer, and presumably the assumption is you have enough of all the nutrients except zinc and oxygen. And that's and one of the arguments for servomyces that people make is that it has a lot of bioavailable zinc. It turns out it's not real easy for organisms, including yourself, to deal with certain kinds of minerals, and bioavailability is a big deal. And apparently servomyces has dead yeast, effectively, in it, and the zinc is in the yeast. And it's in a form that's available to yeast. In fact, in my laboratory, what we grow yeast on is yeast. We grow yeast on a mixture of hydrolyzed yeast, hydrolyzed meat proteins, and glucose. That's our main thing. So they get what they need from that. We also add vitamins and minerals sometimes. So presumably if you oxygenate and add yeast, you'll do okay. Certainly for low gravity worts that don't have a lot of adjuncts in it, because a lot of adjuncts are deficient in some of the vitamins, minerals, and nitrogen and things like that. And when you add them, they dilute out those things in the wort because they aren't adding any of that, but they're taking up part of the body. So I always add some nutrient to my fermentations, but I'm being told here that maybe I don't need to add as much.
So is there a difference between the servomyces and the...
Yeah, so this
nutrient?
is actually, the yeast nutrient is actually a complex mixture of vitamins, minerals, and nitrogen.
Okay.
And then there's DAP, that is nitrogen and phosphate. So phosphate's an important thing, and I believe there's phosphate in here too. There will be some of those things in all this. This isn't just zinc, it's whatever's in the mix. So there's a number of these more complex. I happen to use this thing. There's a number of these more complex ones, and I think it's good to add some, but I sort of got wondered after seeing the idea that if you have too much nutrients, you can encourage overly aggressive growth and maybe more off flavors. But that is me. So that's this thing. So that you can encourage off flavors if your growth is too aggressive. I don't think that's why growth is really so aggressive when you have an aggressive process fermentation process, I think it's mostly because, one, you usually have a really high gravity wort, and two, your temperature is probably not controlled really well inside of your fermenter. Keep going to push something else, but it's in my hand. Okay, so the last thing before we take a break is that I wanted to mention a few things about yeast storage. So you know that you buy packs of yeast from White Yeast or White Labs or wherever, and they have dates on them, and they want to make sure you use them in a certain amount of time, and they're stored in the refrigerator and all that. You know, yeast storage is, you should be concerned about maintaining viability as well as you can. Now, a lot of people want to reuse yeast, and you can easily do that if you do it, don't store your yeast for too long, or if you store your yeast and then you make a starter. So if you can harvest yeast from your fermenter, and you can either wash it or not wash it. You can store yeast under beer if it's a low gravity beer, low alcohol beer. You don't want to store it under a 12% Belgian because alcohol is toxic to yeast, and a lot of yeast can't even tolerate that level. But obviously, if you made a 12% Belgian, they could. But they may not be real happy by the time they get there. So you don't want to store yeast under that, and probably you don't want to reuse real yeast from real high gravity beers. Or you can use sterile degassed or sanitized degassed water. I would boil your water, and boiling degasses the water. You just don't want to get a lot of air in because you don't want a lot of oxygen when you're storing yeast. There's no oxygen in the beer, presumably. And when you boil the water and chill it down, it's mostly oxygen's out. And you can have sufficient viability in the refrigerator for several months like that, store it in mason jars or whatever. And but I would recommend that after 2-3 weeks or so, it's a good idea, unless you're using a huge pitch and you don't care really what's in there, I would make a starter. Because you want to wake your yeast up again. And there might have been a lot of cell. You can actually freeze yeast. And in fact, all of us that work on yeast freeze them in glycerin for long-term preservation. I have yeast strains in my laboratory that are almost 30 years old. Okay, they've been sitting in a minus 80 degrees C freezer. It's really cold in there. Okay. And they are happy when I can scrape a little bit of it off the frozen surface and streak it out on a Petri dish, and I get yeast colonies. Okay, so you can store cells really good in the freezer. There's ways to do it at minus 32. You can do this in a regular freezer, minus 30 C. But it's got to be a non-frost-free freezer, or you've got to somehow deal with the freeze-thaw process that happens when you have a frost-free freezer. Okay, so that's another issue. But you can freeze large samples that are ready to pitch. Sometimes I freeze pitchable samples of yeast and store them for months, and then I take them out and I just make a starter overnight, and they're happy. And then, of course, you can use dry yeast, but we don't know how to dry yeast as individuals, so it's, you buy dry yeast and it's available for you. Okay. And
where do you get glycerin?
You can buy glycerin at Amazon. You can buy them at some drugstores, but you really want food grade. And we call it glycerol in the laboratory, but it's called glycerin when you buy it online. And you can buy liters of it, just like you buy phosphoric acid solutions and things like that. Okay. So we're going to take a break right now. We're going to mix up some off flavors. When you get back here, we're going to taste a couple off flavors, and then we'll talk about the next piece. I don't expect you to read it, but these are all the list of off flavors that I could come up with that are related to either yeast themselves or for fermentation conditions that are associated with the yeast. These are things produced by yeast or that get produced because yeast secrete their precursors or whatever. So we saw acetaldehyde as an intermediate in the formation of ethanol. So if the reaction forming ethanol from acetaldehyde is not efficient, then you're going to get acetaldehyde buildup. That's the green apple thing. I'll just talk about a few of these. Alcohol, we said, was an off flavor. It's only an off flavor when it's way too high in concentration for the beer that you're making. You don't want the alcohol flavor to come through, and you'll see that on judging sheets, right?
Right.
That your beer's hot, okay? It's got too much alcohol in it, or the alcohol is not being hidden by other flavor components that you want to have there. You're all probably familiar or heard of the banana isoamyl acetate, which is an ester that falls really into this category here, but there's lots of different esters. The isoamyl acetate is an off flavor in many beers, but it's exactly what you want when you make a straight ahead Hefeweizen. Okay? Estery, fruity. Fusel alcohols are alcohols, more complex alcohols than ethanol, all fall into the fusel alcohol area. And those things, when they react with acids like amino acids and other things, form esters, okay? And esters are the fruity things. Sometimes they're the solventy things. Then there's these spicy clovey flavors that are also something you want in a Hefeweizen, okay? The compound's eugenol, which is the clove flavor, is a phenol, and there's a whole bunch of phenols again that are off flavors. And then I mentioned earlier sulfur compounds that are produced by all yeast, but they're produced that we taste them a lot in lagers, and I think you'll taste it in this Coors Light. If you just taste the pure Coors Light, then you get these sulfur compounds, which have sort of either a rotten egg, burnt match, cooked vegetable, so DMS, cooked corn flavors are all different sulfur compounds that are produced from breakdown of amino acids or breakdown of all other sulfur compounds. Okay, so there's a whole bunch of these things. And we're going to taste two, but they really cover three categories. The one you have in front of you that you could think about tasting now is diacetyl or diacetyl, both correct pronunciations, okay? It is a compound that results from long lag phases. It's produced in all fermentations, but if you let the fermentation sit for a bit, at the end, it will be metabolized. It's a buttery, slick mouthfeel, and it can be avoided by having healthy yeast, so you don't have a long lag phase, and or by having a post-fermentation rest, or both. And the post-fermentation rest, if you've done lagers before, you always have a diacetyl rest. Okay, it's at the end. You pop up your temperature higher than you've been fermenting at after your beer's fermented out, and let it go a day or two. And that's for re-metabolism of diacetyl. I was going to talk about that on the next slide, but so that's your first thing to taste. The next thing we're going to taste is an ester, which is ethyl acetate. Okay, so it's another one of these acetate esters, but it's not isoamyl. And this one has kind of a fruity flavor. It's present in all fermentations. It has kind of a fruity flavor. I'm going to go on to the next slide. So we're going to taste some spoiled beer that's additionally spoiled by yeast-associated off flavors. We'll start with spoiled Coors Light. So we're tasting this guy now, and Mike is now mixing up. I got a little vial from Mike Cabrat, who got it out of one of the Siebel kits, I guess, and it is this ethyl acetate. And it's fruity at low concentration, but it's solventy at higher concentration, and it has kind of this nail polish remover flavor. And I can tell you that a lot of wild yeast make kind of nasty off flavors that are sort of related to this, although I haven't tasted directly this sample, but I'm supposing that's what it is. And it's actually the most common ester in beer, the most abundant one, but it has a very high tasting threshold. that can be 100 to 1,000 times higher than a lot of other esters that we taste.
So how do you open this? You just pop it?
Oh, you need, we need like a paper towel or something, and then you gotta go full before you,
Just put it over this, maybe?
yeah, but I don't want you to cut your hand, because sometimes they break jagged.
There's those cans up there.
It's lucky one.
I don't want to die.
Okay. So you notice that it has a tasting threshold of 33 parts per million. And most of these esters are sort of in the 10 to 300 parts per billion. Okay? So it's a big difference in its threshold. And hopefully we taste it. And I noticed Mike made a face, so.
You can smell it.
So this will be our other off flavor. And then we'll go on to beer stocks.
The next one.
So you won't die, these are all okay.
Ethyl acetate, yes. Did everyone taste the buttery thing? I think I dosed it pretty good. It definitely tastes different than the parent beer.
I can't taste it.
Huh?
I can't taste it.
Really?
I'm not saying.
Oh, no, did I?
This, all it is imitation butter flavor. I'm gonna give you a bottle, put a couple extra drops in it. I can taste it in this, but I couldn't smell it real strong, but I had it on my hands, so I wasn't sure if I put enough in or too much.
There's like the other
I recently got sensitized to diacetyl because I went to a bar with my son. I went to New York City, to Manhattan, and my son took me to this bar, Proletariat, which you should all go to if you ever get a chance. It's a little dive bar in the East Village, but they have an unbelievable sour and wild beer selection. And I tasted a wild beer, it wasn't really a sour, and I can't remember who made it. And the minute, my daughter actually ordered it, and the minute I put it in my mouth, it was like, oh, imitation butter flavoring. It tasted like the bottle. It was unbelievable, and now I think I'm sort of sensitive to it. And I don't know that I'd ever tasted it before in a beer.
Yeah, we talked about that when you're judging and you have another judge with you. Some people are more sensitive to other stuff and others may not pick up on it, so that's always good. That's why you have two people judge a beer, because maybe someone's more sensitive to diacetyl or diacetyl, however they say that. And so, there's no proper way.
Yeah.
That always drives me crazy. So it's good to know, because I might not pick it up, but someone else may say, wow, I get buttery flavors, and they're more sensitive. So this one, I don't know, do you smell the?
I can get the smell, but the smell isn't real offensive to me.
It's a red one.
And what is the yeast?
This is nail polish remover. This is the ethyl.
This is ethyl acetate. So the reason I said that we're getting 2 in 1 here is because supposedly this is the same flavor of an even greater dose of some of the fusel alcohols.
If you really like inhale deeply, it does kind of burn your nose a little bit.
It's definitely, again, different than the base beer, and it's not incredibly hot to me, but it's definitely there.
Which ones did you want to grab?
I think we should do this. I think 300 to 380.
The 200?
Yeah, and what do you think, we need like 3 pints each?
Sure.
Okay, so now we're going to crank through some more stuff. Everyone getting what they want to get out of this? I don't have any more of the ethyl acetate, but if you didn't get the butter thing, then there's a bottle of imitation butter. You can just put a drop or 2 into your, and so the one I had that was really tainted, you could actually, it just smelled like imitation butter. Which if you smell this stuff, you'll know exactly what I'm talking about. Okay.
The ethyl acetate was like real slight. I mean,
Yeah, I don't.
definitely different.
I asked Mike if this was for, he said this was for a liter, and that we use like 36 ounces, so
Sometimes his kids were of various ages, so sometimes.
Okay. Oh, yeah, that's another point too. I don't know which one this was from.
But if you didn't tell me it was that, I would never want to.
Exactly. So sometimes, so that's the point about this, is that it's desirable in an estery beer at low concentration. So it's not considered an off flavor, despite the fact that if you get too much of it. So, but I agree with you. This isn't offensive to me in any way. Yeah, it's more complex. Okay, so now what we're doing is yeah, I think we're going to need 3.
Yeah, but these are the 300s, and
Oh, okay. So I'm going to go through some beer styles, and we're going to taste some beers, and maybe we should let me see what time it is. Yeah, so we can start spreading these around, and again, keep track of which. I'll just tell you, we're going to taste 2 different Hefeweizen's that are made with different yeast strains. And for those of you who are aware of the White Labs yeast strains, one of them, the first one, I believe, is the 300, WLP 300. The other one is 380. Okay?
Which one is this one?
This one's 300 that's going around. So let's not distribute the next one yet. Okay, so let me talk a little bit about yeast strains and beer styles. Okay, so how do we select yeast strains to emphasize beer characteristics that we want to achieve? So the first thing I just want to talk about is sort of the yeast forward beers. Okay, so there's beers that we avoid having a lot of yeast character in, and there's beers that we are interested in having the yeast character in, and a lot of those yeast characters are what we call off flavors in other beers we expect to be clean. So, you know, this is one of the issues of judging, is that we have to be able to distinguish when these are appropriate. That's why it's really important to understand and read the guidelines, right? Because sometimes it's a good thing. So, I want to go through a couple, a list of sort of yeast, what I'm calling yeast forward beers that require specific strains. So Hefeweizen, which we're going to taste, emphasizes esters and phenols, and in this case, it tends to emphasize the banana E isoamyl acetate and a phenol called eugenol, which is the clovey flavor. It might have other esters in it too, but those are the ones that come through most. And one of the example strains, one of the classic strains, is the WLP 300 or Y Yeast 3638, which are classic Hefeweizen strains. Now there's Hefeweizen strains that are heavier in the banana, and there's those that are heavier in the clove characteristic. 300 is supposed to be heavier in the banana. 380, which we're going to taste as the second one, is heavier, supposed to be heavier in the clove.
Are you guys getting banana in this one? That's not
more close than a banana.
Yeah, that's how I know.
300?
Is it still?
It's still 300.
So I can't
It's
hear
only
the question.
300, yep.
300. Okay.
I can't hear the question.
With F's like bubblegum, would that be
Yeah.
like ester or, you know?
That would be from esters. That would be from a mix, and I don't know what the mix is on those. I don't know. I don't know what people care. I know what bubblegum
Yeah, I
tastes
mean,
like.
I think when they're explaining bubblegum, they're because you're describing it when you're judging, you're describing the beer to someone as if they weren't drinking it. You're like, what does it taste like? And that's, you know, you associate, like if it tastes like marshmallows or if it tastes like Nestle Quick Mix, then those are the terms that you try to use something that someone will know. You don't use the term like, it smells like grandma's closet. Well, who's grandma's closet?
But the other thing is that
Right.
the other thing is though is that we characterize this as a banana flavor, and I don't characterize bubblegum as a banana flavor. So it's a different complex of esters, and it may be one ester or more esters. I don't know. And there are, you know, there's an appropriate dry yeast also. What I did was try to take a selection from each of the. Didn't want to bias everything towards White Labs, you know. So I just tried to get a selection. Another of the sort of yeast-forward beers clearly are the Belgian beers, all the Belgian beers, but the ones you can taste the esters and phenols best in are the ones that have the cleanest wort profiles, that don't have a lot of roasted malts in them and things like that. So I put up Belgian Pales and Belgian Strongs, and those are, again, esters and phenols, but as most of you know, it's a totally different spectrum of flavors, right? And some of them are really phenolic and spicy, but not necessarily a clovey spice, a different kind of peppery thing. You know, you think about a saison or some of the pales, and some of them are going to be estery and fruity, and some are a blend of the two. And these are a couple of the yeast strains I just picked out. Then there are English and Scottish ales that tend to emphasize esters too. And so some of these strains are a little bit more estery than what we call our clean strains, our California ale strains and things like that. And then we get into a whole nother class of things. And of course, this is a whole nother yeast, which is Saccharomyces pastorianus, which is the lagers and pilsners. So these are the Czech pilsners, the American lagers. They have a tendency towards having a lot of sulfur compounds in them. A lot of them are sort of blowing off sulfury smells when you open up the bottle. And for some of the beers, it's not considered an off flavor. For some of the beers, it's a big flaw. You can have some DMS, but you don't want a ton of DMS. Okay? So here are some of the examples of that. Obviously, there's lots of other alternative strains. And then for wild and sour beers, of course, many of those have all these flavors and more. And the big one would be acids. And then for those, we might be using a combination of some of these strains with wild yeast, with Brettanomyces, with bacteria, etc. So there's a whole range of things that can happen there. But you're selecting your organism to emphasize the style of the beer. If you go out there, you can probably find like a wheat beer that's made with Burton ale yeast or with Tally ale yeast or something. It's a whole different deal.
Could you just, this might just be ignorance on my part, but what distinguishes a wild Saccharomyces from not wild?
Well, most of these strains that you buy were once wild strains, but they've been selected out, and some of them have been improved upon by selection, okay? But when I say a wild strain, I mean like strains out of the wild, like you might do an open fermentation. I'll show you a wild strain in a second. I showed you a wild strain earlier that I got out of a wild fermentation from a cider that was just, the apples went into the fermenter and let it grow, okay? So in beer, usually, you're either going to have to encourage bacterial growth, like when you make a Berliner Weiss, like if you do just a sour wort or something. Those are the bugs that are present. Those are all wild. They're just things that came in with the grain. And then you might ferment it with a clean yeast to get the alcohol levels up or something like that. So some strains we refer to as wild, even though we've domesticated them, because they behave like wild strains. They produce flavors like wild strains. So people refer to Brett as a wild yeast. It's not a wild yeast anymore because we've domesticated, at least the strains we buy at White Labs, okay? But it produces that range of flavors. Okay. Oh, and I was meaning to show you some of these are some of the commercial examples that I picked the first one on the list and searched the internet and pulled this picture. Okay, so the first one on the list for the style in the style pack. So that's a half and a golden nail. So then another way you might go about selecting a yeast strain is not to emphasize the yeast, but to emphasize a component of your beer that you want to show off, a component of the wort or the hops. So for instance, we all know that when we want to show off hops, we're going to use 001 or 1056 or SO5, right? So you know, you want to make a grunion, you want to emphasize the hops. That's now the top ranking pale, I guess, or at least the first one in the style guide. I don't know if that means that they thought it was the best one, but it's the one in Sierra Nevada pale anyway. So we choose those kinds. If we want to make a more malt forward beer, then we want something that emphasizes the malt, which tends to be in part because it doesn't attenuate as strongly. So it's not leaving some malt behind. And a lot of the British yeast strains do that. And there's even some lager strains that are really sort of trying to emphasize malt. And one of them is the one we're celebrating right now, which is Oktoberfest style beer. Okay? And these are some of the strains. I don't mean to say what's best and what's worse here, so don't take these home and think, oh, that guy said these are the best of that stuff. I just picked some stuff. Okay? And then some strains, we want to bridge the divide. We want to make a lagery beer, but we want to ferment it with ale yeast. Okay? Or so there are these hybrid style strains like a Kolsch. It's an ale yeast, but fermented at cold temperature and makes a lagery style of a beer. And I think I have one of those. And these are some. This was a selection of a Kolsch and an alt beer from each of the big vendors. So I'm not endorsing any vendor. So that's another way. And then sometimes you actually make a beer of a particular style because you know the yeast will behave well under the conditions you have. So if you wanted to make a beer last week and you don't have any way of controlling fermentation temperature, I hope you made a Belgian. Because it can handle the warm temperatures at least late in the fermentation. Okay, and it emphasizes the things you want to emphasize. There are off flavors in other beers. There are good flavors in this beer. If you want to, if it's cold, you might make a lager or a pilsner, right? It's 50 degrees in the winter where you're doing your fermentation, then you might want to do that. And then I just wanted to make a pitch for experimenting with fermenting with different yeasts. So the best way to do it is just like White Labs do. We're in the mecca of this thing. So you take a wort and you ferment it with a bunch of different yeasts, and then you get to see what the yeast does in that kind of wort. And then you make another wort, you ferment it with the same yeast, or you can ferment it with a group of different yeasts, etc. Almost every time I brew, I'm brewing 10 gallons at a time now, and I split it in half, put it in 2 fermenters, pitch different yeast, sometimes different hop schedules, sometimes I split the boil, sometimes, I do all kinds of stuff. But it's a really good thing to do, or to split off, you make a 5-gallon batch, split off a gallon, and try it with some different yeast and compare it to what you just did, if you're not sure about it. And so you can do that if you're White Labs, you have a bunch of 5-barrel fermenters, and if you walk in the back room here or look through the door, you'll see these guys. I mean, there's a whole slew of 5-barrel fermenters. You can do that. Or you can go to my lab, which is really my house, and I make a thing. You can make liters, gallons, whatever you want. I was doing a really small scale here because I was comparing a bunch of wild cider yeast with a bunch of apple juice. And so I had 10 fermenters, and I just put a blow-off tube on each one. I stuck all the blow-off tubes on a bottle of water in the middle, and you could do 10 1-gallon fermenters, and you could, if you do small volumes, you can put them in your fermenting refrigerator or whatever. Okay, so
So I had a question. So I've done this before. So we brewed 5 gallons, split it up into 5 1-gallon batches. If I pitch an entire vial, is that over pitching, right?
Yeah.
I would say that's over pitching.
So,
How much do you-
so then, yeah, so I'm going to do this 1-gallon experiment. I'm going to go home. How am I going to do that? Am I just
You're gonna
peel off
make
all
up
of
a slurry. You're gonna pour off a little bit of it,
it? Okay.
and then you're gonna put the rest in your refrigerator and use it to make the beer that you thought was best.
Okay.
Dry yeast is awesome.
But I mean, if you want to figure out like the different White Lab,
Yeah.
the weird, the 1,
Yeah.
2 to 3 to 4 to 5,
Collect strains from your friends.
Okay.
Um,
But you don't know how well they.
no, true. I mean, it's a
Handle
dilemma.
these.
It's a
Yeah.
dilemma. Okay. But yeah, you can do this thing that they do at home. They just have a really cool.
I can have all that.
So this is someone referred to, was it Rex? They asked about the hairy eyeball. Oh, so this is the hairy eyeball. This was just to show you that there's other yeast out there. They do crazy stuff. It's not the best image with these lights on, but this is a colony of yeast growing on a petri dish. And normally that would just be a round blob. You've seen the pictures of them. They're just a round blob and they're just sitting there. But this guy, and this happens to be through a microscope, but if you look at it even with your eye, you see that there's a bunch of stuff going on around it because it's growing out all these filaments and it's actually penetrating down into the agar. It runs out of food under that blob. That blob's really dense, so it starts looking for stuff. And if you wash this plate, this would go away, and you can see why I call it the hairy eyeball. But it's this is a strain called Pichia, a genus of yeast called Pichia. It's one of the species, and this yeast this ferments really, really slowly in both cider and in beer. I haven't tasted a beer I've made with it. I just did a little test fermentation. But the cider, it does an interesting thing too, in that it softens the acids. It does malolactic fermentation. And so you can mix it with the Cerevisiae strain. It's going to ferment the alcohol, the sugar to alcohol, and then you get another interesting flavor profile from adding this yeast to it. So
Those aren't filaments, those are actually the cells.
those are strings of cells, and they're actually individual cells, but they never divide from each other. So they all end up in a big chain as they divide. And they're mechanically rigid enough that they dissolve the agar and they can dig down into the agar. Okay?
So they split but not completely?
Right. So where I showed you the bud coming off and the little bud scar, they don't make that bud scar, they just make another one and then they make another one off the end of that, another one. These may divide, but they come out the end, so you get a long film. Okay. So there's a whole bunch of commercially available yeast that we might refer to as wild yeast and bacterial strains to develop complexity in your beer. And some of them, Brettanomyces, which is now actually called Decara, but none of us brewers refer to it as that. And then even some Saccharomyces strains. So you probably know the story that one of the strains that was sold by White Labs as Brettanomyces was the DNA was sequenced and it was determined that it was actually a Saccharomyces strain. Looks like Brett, behaves like Brett, makes the same flavors as Brett, but it's a Saccharomyces, wild Saccharomyces strain. And then there are lots of other strains. Some of them are in the Saccharomyces family, which is what this means, or they're not even Saccharomyces. And if you do these indigenous fermentations, where you just leave your beer open, and if you like things like that, you should read the Milk the Funk web page and their Facebook group, where people are doing crazy stuff like this all the time. They throw some wort out in their backyard and wait for it to start fermenting, and then they see what they get out of it, put it in a barrel, do whatever. You have amazing things going on. And there's really good literature about this, and I've done some work on this where you, like with ciders, where you can follow the different species come up, other species go down over time. And there's a lot of crazy stuff that goes on. Sometimes some part of the fermentation it tastes terrible, some parts it tastes great. There's all kinds of stuff. And these are all these strains, when you either individually or together, depending on the strain, can form all these different characteristic flavors, these barnyard, leather, smoke, people refer to the Brettanomyces character, pineapple sometimes, acids of various sorts. You can enhance acid by using lactobacillus and pediococcus and even acetobacter sometimes, which is what turns wine into vinegar. Okay. All these things are active in certain wild fermentations. Some people pitch them intentionally. So there's lots going on beyond these guys. And I saw this picture on Milk the Funk today. I thought it was the most beautiful pellicle. It's so clean looking. Usually they're so just rankling. So I just put it up here. I actually asked this woman if I could put it up, take her picture and put it up, but she didn't respond, so I put it up anyhow. But I gave her credit. But I thought that was cute. And that's what some of these strains that form, this is mostly bacterial probably, but you get the fungi and the bacteria. This is a true biofilm, if you've heard the term biofilm. You get these biological communities that do crazy things. I gave you some resources. Okay, So that's the end of the talk.
The 380 beer went around.
And
What
when
did you guys get on that? Is that the Clovey? You get banana?
So the banana one is supposed to be the Clovey one, and the Clovey one's supposed to be the banana one, and they were supposedly fermented identically,
Right.
exactly the same temperature with the same worts, and that's the way they're described, is that 300 is supposed to be emphasized banana, and it has clove. 380 is supposed to emphasize the clove and have very little banana. I agree with that completely. I thought exactly the opposite. But that, just for the purposes of this talk, those are pure yeast byproduct flavors. They don't come from anything in the beer. It's a relatively clean wheat wort. Okay?
So one of the things I heard earlier as we were setting up and they were talking in the back, this is the employees, I think they may have a phone number here, and so I don't know if it's in their literature, but you can call in if you're making a beer, and if you want it to do something or taste like something, you can actually talk to them and say, hey, I'm making this beer, I want to have some pineapple flavor. What yeast would kind of produce that? Or I want to do mango, or I want to accent this and that. And from the talk, they said they're here to help. They'll tell you what strain, how it'll work, maybe an optimal temperature. Like if you read Jamil's book, he goes in there and talks about doing a Hefeweizen, and I did it. If you ferment at 62, it neutralizes the banana and the clove, and it's the
That's
weirdest.
what
Yeah, 62 is like nothing. You get nothing, and it's just a wheat beer. So
I thought they did the 300 too cold, is what
Yeah.
I thought probably was the case.
So they'll tell you if you want to ask here, and I don't know if the folks that are working in the bar, they probably would know.
Jimmy knows a lot.
Yeah, so definitely hit them up and ask if you're doing something with your own beers, or if you judge and you're like, yeah, picked up this weird flavor. That was what would cause that.
Was it brewed on their old system or the new one?
Here, the new
Here
one.
in the new one.
The new one. The old one, the other one was 65 degrees.
I think so. They said they only had one fermenter or
Same
something.
room, yeah.
Yeah. No, these are all jacketed fermenters, and they all regulate the temperature, and all of them appropriate to the yeast that they're using, they say. They didn't tell me. I think we can find the recipes for all these things are online. I'm not sure all the fermentation temperatures are online. But I thought it was kind of interesting.
The 300 seems a bit more balanced.
Yeah, do you know how much that was?
I don't.
Do you know how much the pints even are?
I did it.
I don't know. Yeah. Yeah, but if people want to throw a couple bucks each in, it would be great. I don't have change for bucks. So. That all went on my credit card. If you can't throw a couple bucks in, then don't worry about it. I don't mind covering it. Any questions about anything else? The other thing is that we got a lot of cups out today and stuff, so if people want to help a little bit with cleanup.